Good evening, everyone. Today I'll be discussing lysogenic conversion. To start, I'll briefly describe a provirus. So a provirus is an inactive virus in an animal cell. And it's important to note that the latency of animal viruses is different from lysogenicity of phages. Although animal viruses have the same 5 basic steps in their replication pathways as bacteriophages, there's a significant difference in the replication of animal viruses due to the presence of envelopes around some of the viruses and from the eukaryotic nature of animal cells, as well as the lack of a cell wall. However, bacterial viruses interact with their hosts very differently. So some bacteriophages have a modified replication cycle in which infected host cells grow and reproduce normally for many generations before they lyse. This replication cycle is called a lysogenic replication cycle, or lysogeny. Lysogenic replication is different than lytic replication because in lytic replication, the bacteriophage immediately starts replicating and ultimately lyses the host cell, resulting in new viral particles. Lysogenic conversion, on the other hand, is results in a change in phenotype of a bacteria due to insertion of a lysogenic bacteriophage into a bacterial chromosome. This then allows the virus to replicate along with the bacteria's genome without causing death of the cell. Additionally, lysogenic conversion is able to lead to the acquisition of new traits by the bacterial host, which is in contrast to lytic replication. When viral DNA enters the bacterial cell, the host cell's DNA is not destroyed, and the phage's genome does not immediately assume control of the cell. Instead, the virus remains inactive. Such an inactive bacteriophage is called a prophage. A prophage is an inactive bacteriophage which has inserted into the host chromosome. A prophage remains inactive by coding for a protein that suppresses prophage genes. It's important to note that a side effect of this repressor protein is that it renders the bacteria resistant to additional infection by other viruses of the same type. However, every time the cell replicates its infected chromosome, the prophage is also replicated. So therefore, all daughter cells of a lysogenic cell are infected with nearly inactive virus. A prophage and its descendants may remain part of the bacterial chromosomes for generations to come, sometimes even forever. This is different than lytic replication, which involves the direct transmission of viral genetic material from one bacterium to another, rather than passing on genetically through daughter cells during bacterial reproduction. Additionally, lysogenic conversion plays an important role in the development of pathogenicity in bacteria. Bacteriophage genes are responsible for toxins and other disease-invoking proteins found in bacterial agents of diseases such as diphtheria, cholera, rheumatic fever, and severe cases of diarrhea that can be caused by E. coli. Thank you so much, and I hope that cleared up some things about lysogenic conversion.